All right. Thanks, Caroline. So I'm Carice St. Hilaire Clark, and I'm a program officer in the Division of Neuroscience here at NIA. So I'll be moderating session 2, which is going to be, again, focused on air pollution, health impacts, and aging, but we'll also hear a little bit more about noise pollution. In this round, we'll have talks from 4 speakers, which I think are quite amazing. We'll start it off with Dr. Tuck Finch at the University of Southern California, and he'll be followed by Doug Brigger from the University of Connecticut. And next up will be Dr. Jay Pinto from the University of Chicago, and rounding it off will be Dr. Jennifer Weave from Boston University. So Tuck, without further ado, I will give the floor to you.
Okay, thank you. So let me.
During the discussion session. So what we can do right now is just head straight into the next speaker. And that will be Dr. Doug Brigger.
Do you want me to go ahead?
Sure.
Okay, great. Thank you for having me here today. It's great to be here. Really interesting group of speakers on topics that are all relevant and interesting to me. I'm going to talk about ultrafine combustion particles from traffic and our work on cardiovascular biomarkers and then moving into protective measures. I think most of you know what ultrafine.
Do you want to go into presentation mode?
Yes, I do. Sorry about that. Yes,
No
that's
worries.
much better. Thank you. I think most of you know what ultrafine particles are. So in the interest of time, I'll skip this slide. I also think you know that ultrafines are a major source of ultrafines is from road traffic. And this is from the European Union and from the United States, and one of the top 2 sources. There's been some discussion about pathways. I just thought I would point out, this is from the US EPA Integrated Science Assessment, but these are the 2 pathways we have focused on, which is autonomic nervous system through blood pressure. Blood pressure is a key outcome that we measure. And then we have not done arrhythmia or heart rate variability, and we do very little work on heart outcomes, but this is where we are on the autonomic pathway. And then even more so on systemic inflammation, misspeaking here, and then leading to heart disease, myocardial infarction, stroke. And again, we don't have much outcome here. We're mostly focused on the biomarkers. It's been mentioned before, and I'll just emphasize it again. And this is the emerging area, and it's a theme in this session that I think is highly important. These ultrafines can go directly into the brain via the olfactory nerve. In my opinion, that evidence became convincing a number of years ago. Lillian Calderon Garciduenas in Montana has been a leading person in this, and I would just point you to this very nice photomicrograph, transmission electron photomicrograph showing ultrafine particles in mitochondria and brain tissue at autopsy that she very kindly included in a chapter in the book that Christina Fuller and I did put out a couple of years ago. And so I think that's a very important direction. We haven't worked that much on the neurological effects yet, but these ultrafines are traveling through the body, crossing biological barriers, and we think that is very important. Another colleague of mine, Mei Chung, took the lead on a systematic scoping review that we published a number of years ago of ultrafines and ultrafine epidemiology. And this ended up with 97 studies. Already this is out of date, which is of course a problem with systematic review. This only goes up through 2017. But May did these very nice bubble maps. I think this is a methodology that she pioneered. And each bubble is an outcome from a study. The red is statistically significant. The yellow or orange is a trend that doesn't reach statistical significance. And green is no evidence of an association. And then the size of the bubbles are the size of the study sample. And you can see here we have cardiovascular outcomes, respiratory, and then other. And I think what you can see is there's a lot in cardiovascular, less in respiratory, and even less in other outcomes, which is a gap. The 2017 cutoff, obviously, there have been many more studies since then, and this kind of work needs to be updated at some point. The other thing is that all of these are short-term outcomes. Long-term association studies with ultrafines are much rarer, although again, there have been more since 2017. The long-term outcomes that we had studied are mostly here. And I'm going to talk a little bit more about that work next. What we found in our first study of ultrafines and the primary outcome we were looking at were blood biomarkers of inflammation, was that we had to get very fine grain with our exposure assessment to see the effect. And so what this is a slide that Kevin Lane, now at BU, who did his PhD on this with us, did. And what it shows is if you just do address matching, you can be way off, as much as, in this case, 85 meters off, here 90 meters, 68 meters. Just the location of the residence could be wrong, especially in large housing developments. And so the first thing that Kevin did was, so we're working with much smaller numbers. It's 700 people enroll in the study, a little over 400 with complete data and biomarkers for the main analysis. But he went in and adjusted each one to their apartment within the building in order to get a really fine-grained assignment of their location. And then the other thing that Kevin did, which was also critical, was to adjust for their time activity behavior. And I won't go into in detail here, but just to show you that time at home in the red, time at work or school in the purple, people who are employed versus people who are retired or not employed, very different time activity patterns. The black line is time spent on highways. And when you make that adjustment, if you look over here on the right, and you have all these high level initial residential exposures near the highway, when you adjust for the time they're away from the highway, those their assigned exposures change substantially. And in fact, in order to see a monotonic association with ultrafine exposure, you have to do this adjustment in our hands anyway. And then in addition, there is an additional step of controlling for confounding. We had negative confounding in this study, and this is changing for time activity, but then there's also an adjustment for confounding in order to really see the effect. And the most recent, which I'll just put up here because it just appeared online in Science of the Total Environment, is that Kevin eventually got around to finishing up his structural equation analysis and gets even stronger associations between the time activity adjusted particle number concentration as a measure of traffic related air pollution. We also included distance to highway in this construct. But when you conclude all three inflammatory markers, you get a much stronger effect of the ultrafine exposure on inflammation. So all the steps along the way, the gold standard geocoding, then the correction for time activity, correction for confounding, and then doing structural equation modeling rather than simple regression analysis, all of those strengthened and improved the level of association that we found. We think that underlying this is the inflammasome. Wig Zamor spoke a little earlier, and he's pulled me into thinking about inflammasomes. I won't go into detail here, but the NLRP3 inflammasome in particular, we think is the way that these particles are driving inflammation and generating IL-1 beta, 18, and then subsequently down there to CRP and so forth. We moved, because we do community-based participatory research, we moved into looking for protective measures early on. This is a review that came out recently of controlled studies, randomized studies of air purifiers in homes. We did 2 pilot studies that are included here. Overall, the evidence suggests benefits. This is on, let me see if I get it right. The top is C-reactive protein, and panel B is interleukin-6. So, so there's some evidence, but we thought our pilot studies were not strong enough. We did yet another pilot study where a highly controlled exposure scenario. This was published in Hypertension early in 2021. We put people in a room near the junction of 2 major highways in Boston Chinatown, and we were able, and just because people are talking about noise, we put noise canceling headphones on them. We had them sit quietly for 2 hours. They're exposed to low, medium, and high amounts of traffic pollution allowed into the room using air purifiers and opening windows and doors. And what we saw was a gradation of blood pressure from low to medium to high that corresponded with the exposures. And that made us feel fairly confident that if you could really get the air purifiers to work effectively, that you could improve blood pressure.
You have 1 minute remaining.
Okay, I'm almost done. Thank you. And that led us to this randomized trial that we're in the middle of right now. We have over 90 participants that have enrolled, 75 have completed. The first year was very weak. It was the first year of COVID, and we were really challenged to enroll people, but we've picked up. We're doing a lot better now. We have 1 month of either real or sham filtration, and then a month of washout period, and then a month of the reverse configuration with participants blinded to which they're receiving. And we are doing blood pressure, including central blood pressure, and taking blood samples that will be analyzed for inflammatory biomarkers, probably coagulatory biomarkers, and probably metabolome as well, and maybe some other things. We'll have to see what our funding allows at the end. And then I would just mention in passing as well that we have worked at a community level with architects and urban planners and city governments around how to protect near highway neighborhoods. And a lot of our work, especially in the city of Somerville, has focused on the idea of putting noise barriers along the wall. I saw that Rich Baldoff was in the audience, and Rich has been an inspiration for some of this. So that's what we've been doing. We have a website, all our papers are there, policy papers, all kinds of things on our website. And if you just put CAFEH into Google, it should come up as your first site. So thank you. I think I made it.
You did. Thanks, Doug. Okay, so we have time for at least 1 question. There's one in here from Wig. Wig, do you want to ask your question live? Caroline can unmute you.
It's odd for Wig to ask me a question. We meet almost every week.
Well, I kind of wanted to make a comment. Happy to have Doug respond or Dr. Finch.
Wig, your audio is a little low. Can you speak?
Okay, yeah, I can up it. Shoot. Is this better?
Perfect.
Yeah.
Okay. Yeah, so as Doug mentioned, I kind of drift around in the literature, and I've been focused on NLRP3 inflammasome for the last 5 years. And I'm glad to see Dr. Finch here because their NF-kappa beta assays with the USC group and Costas Chiotis is just one step before the generation of pro-NLRP3 and the IL-1 cytokines, which are basal to all of this inflammation. And I want to magnify attention to one thing. You know, we had a problem with the diesel truck engines because all the emissions chips turned out to be contraband. That was fixed. More recently, we've had some other emissions problems with VW diesel. NIST, federal reference DEP, underreacts extremely compared to real world particles from highways, so that it's in some cases only 2% as reactive as the actual particles in ambient air when you're doing tests of inflammation. And what that means is the immune cell biologists who live in a different world from the environmental health scientists have been using diesel reference DEP without knowing how unreliable it is. And so they've concluded that real world ambient particles, which are as variable as all manufactured particles combined, are divorced from the basic inflammatory particle pathways that have evolved over 1 billion years. And I'm afraid that's completely untrue, and that it won't be uncovered without facing this federal reference DEP problem.
Thanks, that's very informative. All right, well, I guess.
No, you're muted. I guess you're muted, Todd.
Can I be heard?
Yep.
Okay. May I comment that the NIST DEP have almost no activity in the in vitro assays, but they're highly active and reproducible in vivo. That's my experience. Lung and brain. But in vitro, they're not reactive. They have to be physiologically ingested by cells.
Right. Thanks, and we could talk a little bit more about that in the discussion as well. So
Good.
let's, oh, do you want to say something else then?
No.
No, not for now. Okay, so let's move right along to Dr. Pinto. Oh, there you are. I was just looking for you on the screen. All right.
There is my
Oh, here
talk
it is.
here. Okay, give me 1 second. There's my window, sorry. Okay. Sorry, I'm just going to put this on full screen.
So if you do Control L, it'll put it on full screen mode.
Thank you. Great. Okay, can everybody see that? That should be good.
Yep, that's good.
Great. Well, thanks for inviting me. This has been a really interesting series of talks. I'm going to talk about the olfactory system as a pathway for air pollutants into the brain. So I'm just going to start with a little background about the sense of smell. So the sense of smell is a little bit of an underappreciated sense, but you can get a magnitude of how important it is to people by how many dollars they spend on candles. So this is a $70 French candle that people buy. And the candle market in the US is $4.2 billion a year. And that just sort of struck me as like, you know, fragrance, odor is really important to people. And, you know, I think the sense of smell, like hearing and vision and some of the other classic senses, really allows us to interact with the world around us. And that's why it has a really important impact on our health and our well-being and our social interaction. And, you know, there's a lot of reasons why that sense of smell is important to humans. It's involved in perceiving pleasure. It mediates the sense of flavor, so enjoying food. And it also allows us to detect danger in the environment. So smoke, spoiled food, you know, warning signals in our environment. It's very important in memory. That's why it has a very close connection with neurodegenerative diseases. And it's very involved in emotions and mood. And these are all critical parts of our lives. And in the cartoon you can see here, part of the reason for that is that the olfactory nerve is directly connected to all of the areas of the brain that I was just mentioning, including the amygdala, the entorhinal cortex, the orbital frontal cortex. So really closely connected to these sort of core human functions. And indeed, the nose is a portal to the brain. The olfactory nerve crosses from the nose, where it's involved in detecting the environment. It's an environmental sensor. And those branches of the olfactory nerve go through little holes in the skull and enter the brain directly. They then synapse in the olfactory bulb and then go on to all those areas that I showed you on the page before. And so this provides a potential pathway for airborne material to enter the central nervous system. There's evidence of this in animal models. There's evidence of this in humans, as was mentioned just before. And so the question, we're all thinking about is how do pollutants affect the brain and aging? And, could this be a pathway that affects olfactory aging, so injury to the olfactory nerve and the sense of smell, and then also related to brain function? And, you can think about this pathway, from nasal olfactory mucosa into the olfactory bulb, into the olfactory tract, into the portions of the olfactory cortex, and these things are connected to other parts of the brain. So you could imagine that there could be a pathway here from nose to brain, and what we think about is sort of both olfactory specific regions and olfactory specific structures. And I'm going to show you some work on the olfactory bulb, but we also think this could be also connected to other parts of the brain, what we are non-neurologists refer to as the deeper parts of the brain. So this is work in our project AeroNose, which is air pollution and Alzheimer's disease, neuropathologic and olfactory mechanisms in a multi-ethnic longitudinal cohort, dementia. Jennifer's going to speak next. She's talking about noise, but all of this work is work we've done together. And we have a nice group of partners. This is, one nice thing about this work is for all of us is that it's super multidisciplinary. So we're working with the team at Rush University, David Bennett in particular, and his colleagues, Lisa Barnes and David Ramirez. Also our colleagues in neuropathology there. Joel and his colleagues and others are helping with air pollution exposures. And then I'm going to show you some work that Ushi Graham and Gunther Oberstdorster have done in looking at the toxicology and imaging of these particles in the olfactory bulb. So this is sort of our model. We've been talking about this throughout all the talks that on air pollution anyway, is you've got this exposure of air pollution generating ultrafine particles. They can get into the olfactory bulb where they can cause pathologic mechanisms both locally there and potentially in other parts of the brain. This can result in olfactory dysfunction and olfactory decline, and also relate to cognitive dysfunction, cognitive decline, and dementia. So the aims of our study, we have an epidemiologic component where we're using Joel's models to map pollution exposure for our participants in these Rush Alzheimer's Disease Center cohorts, which you see at the bottom of the screen. We're testing the hypothesis that higher exposure associated with smell loss and Alzheimer's disease and dementia-related outcomes. These are all longitudinal studies started in as early as 1994, and as recently as 2015 for some of our minority cohorts. So we'll be able to see what the outcomes are over time. And then what I'm going to show you here today is some of the early work we have quantifying some of these particles and characterizing them in the actual human olfactory bulb using some advanced imaging techniques. I should mention all of these cohorts, the patients are the subjects, I should say, are consented for autopsy where specimens are biobanked. And so we've been able to use these olfactory bulb specimens in the analysis I'm about to show you. So, we have some very kind of simple objectives here. We want to know what types of particles get up into the olfactory bulb. Do these nanoparticles interact with cells there, including, do they cause local inflammation? Are they bioprocessed? And then our larger questions are, do higher exposure to these nanoparticles increase neurodegeneration? And potentially, do they get to other regions? And we hypothesize that higher levels of exposure end up with more ultrafine particles in the olfactory bulb, and then are more likely to cause these problems with smell and ADRD. So Ushi Graham is just a wonderful chemist and physicist. She uses high-angle annular dark field microscopy to observe particle location and behavior down to 1 nanometer scale using instruments like you see here on the right. She also does energy dispersive spectroscopy and electron energy loss spectroscopy. So we can also know what does it look like, but what's the chemistry of those particles. And so we can answer the questions of how many particles are there, what are they made of, and how are they interacting with the tissue. So these are what these particles look like. This is an olfactory bulb specimen, and if you zoom in here down to the 200 nanometer level, you see these combustion-related particles, spheres, and they have carbon coatings around them. So we can measure and count these particles in the tissue, and we can see, what she's really interested in, we're all really interested is what does the structure of these particles look like? So they take different shapes and forms. You can form these sort of linear agglomerations in some cases, and other types of physical structures around the cells and inside the cells and causing different configurations. So for example, here you see these kind of big clusters of these particles. And then we can see, using some of the spectroscopic methods, what are these particles made out of? So we can see here, and interestingly, we found a lot of these particles are made of, have components of lead and things like chromium, things that are not supposed to be in the brain. Remember, these are older adults who have lived through gasoline with leaded gasoline. And so we think that, lead is playing a role here. And it's significantly a significant component of these ultrafine particles. So we can learn something about the chemistries and the different types of molecules that are in these particles. We also can see some evidence of reaction to the tissue. So this is a slide showing some evidence of potential nanoparticle deleterious effects on mitochondria within these cells. Again, you can see that some of these have chromium and lead and arsenic and aluminum and silica particles. And we also see some evidence of reaction of the tissue to those particles in the form of ferritin particles. And I'll talk to you about that in a second.
1 minute remaining.
So, thank you. So here we see these particles, and then you see this reaction. These smaller red dots here are ferritin particles that are forming endogenous inflammatory responses to the particulate matter, and we can characterize that chemically. So we can see these endogenous metals, and we can also see here evidence of coatings, so carbon coatings on the surface. So we see these hydrocarbons on the surface of these particles, and potentially we think that these hydrocarbons are potentially reactive with the tissue as well. I'm going to skip that slide just for time. So we think that these carbon or reactive carbon coatings on these metal oxide ultrafine particles are key for sort of toxicity and potentially on mechanisms like many of the other speakers have discussed earlier. Finally, how does this correlate with things that might be important clinically? So we're ramping up these analyses. We've done a fair number of olfactory bulbs so far, and we're also recovering from pandemic and hope to scale this up to our goal, which is to get to about 400 olfactory bulbs, and then we'll be able to look at some of the markers. So in these cohorts, we have pathologic measures, and these are just some early signs that if you look at correlation for the sort of particle amounts and some of these measures that we have in these subjects, like tau density or Braque score, amyloid, and mind you, these are very small numbers and very crude analyses, but you see some correlation with pathology related to Alzheimer's disease and related dementias. So I'll stop there by just saying that, traffic-related pollutants are present in the olfactory bulb. We can quantify and characterize them. They have surfaces of reactive carbon, which may interact with the tissue and result in neuroinflammation. I should mention that we also are looking at effects of these particles on microglia and activation of microglia, so we'll be able to see if that's one mechanism. We think these carbon coatings are important, and we're hoping to show that density of ultrafine particles correlate with AD neuropathology and some of these other outcomes. Lastly, I'll just mention that we're also looking at some other regions of the brain, hopefully be able to show that these particles are progressing from the olfactory bulb to these other connected areas, and giving us further evidence that the nose is a gateway to the brain. And I'll stop there I just want to acknowledge Jennifer, who's going to speak next, who's my partner, David Bennett, who is generous with his expertise and cohorts with his other colleagues, and then Joel and his team, and Adam, who have been wonderful in terms of helping us with the exposures and really think through the history of these people and geocoding their addresses and figuring out what their exposures were over time. And Gunter and Ushi are just wonderful thinkers and helped us with the imaging and the analysis. And I'll stop there. Thank you.
Excellent. Thanks, Jay. That was awesome. We do have a question, but I think that pertains to the previous talk. So we can save that one from WIG for the discussion. I don't see any other questions, but Jay, I was curious. So have you identified the cell types in which you're finding this particulate matter? Is it the neurons? Is it the support cells?
Yeah, so we're in the process of doing that. I'll say that, one thing we've learned is they're a little bit distributed throughout the tissue. So we see them inside the neurons, we see them outside the neurons, and we're trying to work with our colleagues at Rush to sort of distinguish the cellular distortions and distribution of these particles. But yes, they are in the neurons themselves also.
Well, it's a little scary. Any other questions? Chuck?
Yes, that was a wonderful presentation, Jay.
Thank you.
You've cited one of our papers, Hank Cheng, 2000 EHP 2016, which we did a short-term exposure to nanoparticles and showed reaction of glia in the olfactory nucleus. We also have in that paper the comment that after 8 weeks of exposure, the posterior parts of the brain, cerebellum and posterior cortex, have the same level of inflammation response that the front part of the brain, including the olfactory bulb, has, which to us makes us think that the lung also has a major contribution indirectly to the brain, the inhaled particles that reach the lung, because it's hard to imagine how the 3 synapses required to go from the olfactory bulb to the cerebellum would have the same strength of effect in the cerebellum as in the olfactory bulb. So that's, I think we, with full confidence in your demonstration in the olfactory bulb, and I look forward to data and concerning the cerebellum and the brainstem, which I think you'll be able to provide to see if the levels of particles, how they compare between anterior and posterior, near nose and distant nose regions. But it's
I love it that you make the brain just nose is the center of the brain. Yeah, I agree entirely. There certainly can be systemic distribution through the bloodstream. And one of the ways we're trying to answer that is to show that in regions that are connected directly, closer and directly connected to the olfactory bulb, have higher deposition than, for example, ones that are further away or not connected. And we have some controls, and we're hoping to show that. I do expect that there will be a systemic component as well. And we are in fact, looking into comparing deposition in lung also, you know, with the idea that, you know, we just have this one snapshot in all these places. We'd love to sort of watch those particles go and see what happens to them in real time.
It is definitive what you're doing.
Thanks.
All right, great. So we have our last speaker up. Thanks, Jay. Jennifer, take it away.
Does that look okay, or can you see all my tutor notes?
No, it's great.
Okay, great. All right, so I will be shifting back to noise, and off we go. So we all know at this point that noise is an unwanted or harmful sound. And to the right, I show typical noise levels in A-weighted decibels, some familiar noise sources. So for example, a quiet room chimes in at 28 to 33 decibels. A motorcycle roars in at 90 decibels, and rock concerts pummel the ears at 140. Now, as we know, traffic is a major source of exposure to noise, whether from cars, trucks, motorcycles, trains, or aircraft. And noise is also a major public health concern. In the US, that's partially because it's not strictly regulated, as you've heard, but it's also because so many people in the US are regularly exposed to noise at levels that could affect their well-being. So let's take noise exposures that average out to 70 decibels over the course of a year. Yes, that's the noise of a passenger plane cabin representing this annual average. So noise at this level or louder is associated with hearing loss. And in 2013, an estimated 104 million Americans were exposed to noise of at least this level or louder. But adverse health effects have been observed with regular exposures to lower noise levels. Annual noise exposure of at least 55 decibels is associated with cardiovascular and other so-called non-acoustic effects. In 2013, an estimated 145 million Americans were exposed to noise at this level or louder. Now, how might noise exposure influence dementia risk? We heard a little bit earlier about this, and I'm going to just sort of reiterate and add some things. So studies of animals have demonstrated noise effects that are relevant to dementia. So in the hippocampus of rats, noise exposure resulted in signs of oxidative stress, inflammation, and as well as the production of amyloid beta and hyperphosphorylated tau and neurofibrillary tangles. Noise also induced degenerative changes to the structure of synapses, resulting in neuronal apoptosis. And noise-exposed mice show impairments in learning and memory. And by the way, the effects of noise on learning have long been established in children. Now, apart from these direct neurologic effects, it's worth considering that noise seems to influence the development of conditions that, in turn, heighten dementia risk. These include cardiovascular disease and hypertension, diabetes, disordered sleep, and symptoms of anxiety and depression. Now, I also want to point out and just echo what Casey said, that in the US, noise exposure is a matter of environmental injustice. That is to say, community noise is imposed on some communities more than others, and those communities exposed to more noise bear the characteristics of people with less socioeconomic power and less political power. So for instance, as you saw in the US, noise exposures are higher in Black and Latino communities than in non-Latino white communities. They're higher in more racially segregated communities regardless of race, and they're higher among households with low incomes. And these inequities in noise exposure and other environmental exposures merit our attention because they are relevant to who is made vulnerable to developing dementia. I should also mention, we see these patterns even at very hyper-local levels as well, not just with national models. So animal studies are one thing. What do we see in the epidemiologic studies so far? We saw one big example at the front, and I'm going to sort of fill in the details on the others. Now, this picture shows the cognitive trajectories of 3 hypothetical people over the course of their lives. And I'm using this to illustrate the major outcomes in the noise studies that I'm reviewing here. There were studies of cognitive functioning at a single point in time. There were studies that compared the rate at which individuals' cognitive abilities declined. And there were studies of dementia itself. And along with these studies were studies of brain MRI offering comparisons of total and region-specific brain volumes. So I'll summarize the characteristics and findings of the 11 papers that estimated effects of community noise exposure on these dementia-related outcomes. Now, 4 compared cognitive level across noise exposure. 1 looked at rate of cognitive decline. 6 looked at dementia, and 2 at mild cognitive impairment, this sort of intermediate stage, and 2 evaluated brain imaging outcomes. Now, all of these studies used measures of exposure to noise from community sources. So these are not inside people's homes, they're not headphones. And so these sources typically involve transportation, but community noise can certainly come from other sources like construction. Now, 6 of the studies used measures that were specific to noise from transportation sources, typically roadways and occasionally also railways. 5 studies used measures specific to noise from roads, and 1 study used a measure of that was specific to railway noise, I should say, and that was the study of Dr. Cantunaria. Now, all the studies used measures that incorporated noise throughout a typical 24-hour period, so both day and nighttime noise combined. 5 studies also used measures specific to nighttime noise, which could be relevant to sleep and stress responses. Finally, all of these studies aim to measure noise exposure over the long term. And as surrogates of long-term exposure, most studies use noise levels around the time of study baseline. But 3 studies had access to noise predictions that permitted direct estimation of exposures over intervals of 5 years or longer. Now, after all this build up, I'm guessing you want to know what they found. Well, first, let's talk about the studies comparing cognitive performance by level of noise exposure. 3 of the 4 studies reported at least 1 adverse association of noise with cognitive performance. Now, these findings were mirrored in the studies of dementia and mild cognitive impairment. Specifically, adverse associations were reported by 4 of the 6 dementia studies and by both studies of MCI. Now, by contrast, the single study of cognitive decline found no association with noise exposure. And finally, the 2 studies of brain imaging entailed a mix of findings, but I should note that both studies were from the same cohort and assessed mutually exclusive sets of outcomes. Now, for a line of inquiry that's in its toddler stage, the literature boasts several overall strengths, and some of those strengths involve the measures of noise exposure. I previously mentioned the measures of long-term exposure. In some studies, as you've heard about, even determined the orientation of a person's home to traffic noise and incorporated that information into their exposure determinations. Now, many of the studies were able to adjust for numerous plausible sources of confounding, including exposure to air pollution and even green space, as we heard earlier. They were also able to adjust, in many cases, for measures of socioeconomic position. Still, we're just getting started, and it's worth reflecting on how to advance this evidence in both in terms of how we design our studies and what questions we ask. So, first, most of the research on noise and dementia-related outcomes has taken place in Europe or the UK. So there is tremendous opportunity for discovery and impact outside of this region, particularly given different built, automotive, and regulatory environments. Second, we can do more to ensure we've accounted for plausible sources of confounding, especially by factors that have a spatial dimension. This might be air pollution, and granted, I will say some of these studies, when they adjusted for air pollution, did observe attenuated associations, although they were still adverse. We could probably do a bit better on measuring socioeconomic indices as well. And as you saw, Studies of cognitive decline are rare, but this outcome is important because it reflects the very process of deteriorating function.
1 minute remaining.
Sure. And then you know, we can continue to advance this research by applying insights and methods that are well established in dementia epidemiology research, and that includes methods related to misclassification of healthcare data. For example, even in environments where there is universal insurance, the positive predictive value of a dementia record or record of being diagnosed is not surprising to see a positive predictive value of 60%. Likewise, we can start paying more attention to the possibility of survival bias and selection bias, the latter which might be of concern in studies of brain imaging. And I refer people to the Melodem initiative for more insights and input on that topic. Now, in terms of the questions we ask, I'm going to echo something stated earlier in this symposium, which concerns the specific features of traffic noise that might matter. And I want to point to aircraft noise. There has been quite a bit of research on aircraft noise in relation to other outcomes, but nothing related to dementia yet. And our group is currently trying to do this. And specifically, with respect to aircraft noise or other noise sources, it may be worth considering whether we should be using long-term averages or assess intervals of loudness. And as an example, I'm going to show you the noise contours around Boston Logan Airport. Pay attention to this orange line out here. This is the 60 decibel line. It so turns out that communities outside that line have organized around noise complaints related to the airport. And it seems odd that they would be complaining, right, given this map, but it turns out that they have periodic onslaughts of aircraft noise that are not represented in this long-term average. And so that brings me to this idea of engaging with communities about potential sources we may be missing, sources of noise, I should say. Tuck told us that it really made a compelling case for looking at the interplay of noise and air pollution. And finally, I want to end with encouraging the crowd to consider assessing the effects of noise on physical disability, which is an incredibly important outcome to older adults because it affects how long you can remain independent in your own home. And so I've left you with all the papers that I've reviewed here, and I'm open for questions now. Did I get to the deadline on time?
Yeah, you did awesome. Thanks. I don't see any questions in the chat. I don't know if anyone has anything they would like to ask right now. I can. So Jennifer, I was just curious. So for the study, so there was one of the studies where there was no relationship between noise exposure and cognitive decline, I think. What was the geographic location about?
That is a really good question. Give me 1 second. Actually, no, I know which study that. Let's see. So I can give you one example where there was no association. I was pretty generous, by the way, in defining what might be an adverse association. You can see it on the high level. There was a study based in Sweden. The last author's name is, or the first author's last name is Anderson, and no association with dementia. I believe that was the outcome. And when you look carefully at that study, you notice that the range of exposure really tends to occupy the lower decibel part of the spectrum. And so it just may be in those cases that there just weren't people who were exposed at high enough levels. I will say, by contrast, and we're not the only ones here, but in the Chicago Health and Aging Project, with work that I did with Sarah Adar, we had a much larger range of exposures, and we did see an adverse effect. with respect to dementia and with mild cognitive impairment. So it isn't that the first that I mentioned that they demonstrate that there's nothing going on. It's just that they may not have had the range of exposure to see something.
Okay, we have something from Doug. My own experience is that the nature, not just the sound pressure level and pattern of noise, affect how bothersome it is to me. Could you say something about the ability to study this level of detail?
Oh, you mean like noise annoyance, I guess. Yeah, so there's a really nice paper out of Germany. This is, I think, the Heinz-Nixdorf study, probably, that measured both annoyance and noise. And I confess, I can't remember what they found with the annoyance piece. I think that there was a correspondence with noise, but I can't remember what they found with respect to the cognitive outcome. It probably is worth going that route, with the caveat that we also have hearing loss as a common phenomenon in older adults. So, so there may be a lack of annoyance because one can't hear it. It will depend on the nature of the noise, obviously.
Okay, so we can open up questions to anyone at this point. We'll just move directly into our discussion session. Tuck, did you have a question? Yes.
So this has to do with all of the talks today and tomorrow, concerning volatile organic carbons, which are recognized as part of the air pollution, independent as well as together with particles. And no talk today mentioned, and I would be delighted if tomorrow there was some mention of it, but it deserves, it's not obvious. It deserves more attention because of the levels of toxicity that this can induce, both directly in the nose and the brain, as well as in the lung with systemic effects. I think that I just wanted to identify that as something that merits much more attention than has been possible for the field to give so far.
Think something about, oh, this is from Jeanette. There's also the idea of the noise we feel, so low frequency noise. Jeanette, did you want to expand a little bit on that?
So yeah, so sometimes even when someone can't hear, they can feel the vibration that noise has. So people who, for example, have deaf animals, when a car is rumbling by, the dog still perks up. So a lot of people have been thinking about studying that sort of idea of low frequency noise, that rumble, that feeling of noise that is also might be a factor in people's health and quality of life.
All right, so there was a mention from Wig. Wig, there's a paper that you were making reference to. Do you want to unmute yourself and tell us all about that?
Yeah, can you hear me?
Yeah.
Okay, let me go back and look at the.
Your
yeah,
audio went down
this,
again.
yeah, no, I got it. It's just a more recent paper from Kostas Chiotis' group.
It's hard to hear you, Wig.
Okay, hold on. Sorry. Yeah, so this is just a 2021 paper from Kostas Chiotis' group. The first author's Farahani, and it kind of systematically compares standard diesel emission particles, federal reference, to Los Angeles air pollution. And so that's an interesting follow on to all the work that Dr. Finch has helped with out there on the issue of air pollution. If I could, I would throw in a noise comment as well. I don't know if people have noticed, but the US in the last 2 years has a new systematic neighborhood environmental survey on aviation noise, done by some really qualified scientists out of BB&N in Boston. And they're showing much higher noise sensitivity than Schultz did. And Schultz's work was encoded in 1992 FICON decision, where 65 DNL had 12.5% of the population highly annoyed. The NES study shows 20% highly annoyed at much lower levels, closer to 50 DNL. FAA is really perplexed as to how to handle this, because the same strategies and tactics won't work with that much population affected. And I mentioned this in part because Jennifer just showed a relevant slide of the kind of Mystic River and the 33 left departure corridor.
Oh. And Jennifer also just dropped a link in the chat. Wait.
Yeah, wait, can you check that? I think that's what you're referring to.
All right, Chuck.
I'll pass on this.
All right. So we have one from Jay. Are there data about noise experience inside the home? There's lots of data showing indoor air pollution exposures are really high and important. Anybody have details about that?
Jay, I can answer that. there's a small but growing number of studies that have started to look at indoor air pollution, and it's still pretty nascent. I mean, we compare the number of papers on the outdoor pollution, where I think, depending on what you count as a paper, maybe we're talking 70, 75. The indoor air pollution brain data count is probably like 5, maybe. And they generally find adverse effects.
And what about indoor noise exposure? So, some of the things we've been talking about so far are, modeled at the front of the home, the back of the home, day and night. But what about inside the home? I think it's also relevant to think about for the comment about the kind of noise and the shape of the noise. So I can imagine some very noisy homes that have televisions running, or, machines, washing machines, dryer, other things. And maybe there's different kinds of noise inside the house and different levels inside the house.
Like my house. Lots of noise. Okay, so we have Lucy, then Jeanette.
Thanks. I just have a question about the relationship between hearing loss and noise pollution effects. Do you need to be able to hear the noise to be affected by noise pollution? Because it occurs to me that particularly in the case of indoor noise pollution, people with hearing loss have their TVs turned up higher to hear them. And so they may actually be paradoxically more adversely affected than those without hearing loss if being able to perceive the sound doesn't matter in the context of the damaging effects of noise pollution. Did I articulate that clearly, or does anyone know? Has anyone ever looked?
I don't know. I mean, we're talking about ear cells being damaged. So I just, I don't know if any additional noise would further.
I think the mechanical effects on the sensory system may still be there, even though there's hearing loss.
That's a good question. Okay, Jeanette?
Yeah, I just wanted to point out that Matthias Basner is starting to do studies on noise and health effects with monitors inside people's homes. They're trying to pinpoint aircraft noise, but of course they're getting all the other noises. And so they are realizing, as Dr. Pinto said, that sometimes people have their TVs on all night. And so there's a lot that's going on when you look at what's happening inside a home. So it'd be interesting to see the kind of results he gets looking at nighttime indoor noise.
Okay. Let's see, any more questions? Nothing else in the chat? Oh, there's one. I missed this one. A comment from Leanne Shepherd. Inside the home, noise exposure is not that likely to be correlated with ambient noise. So even if this exposure is an important determinant of the outcome, it is unlikely to confound the association with ambient noise. Okay. Thanks, Leanne.
That's super useful, Leanne. Do we just cite you as a personal communication when we write this in our papers?
And
That
to
is
be
really-
precise, I think she's referring to noise exposures that have their sources inside the house are unlikely to, which is different than the question about how much of the noise from outside is getting in. Right? So.
I want to make if you don't mind, I want to make a final plug for more studies outside of the UK and Europe. And I think hearing some of the comments about the policy environment in Europe helps to explain why it's possible to do that research in Europe and why it's difficult to do it here. And so one of the things that I have noticed is in reviewing these studies is the correlation between air pollution exposure and noise exposure in different environments, and it actually varies depending on setting. And I think this is an important thing for us to be looking at for all manner of reasons, including potential confounding and the capacity to evaluate different combinations of exposures. If we have two exposures that are pretty collinear, I should say, it's going to be really challenging to do that. But I think that there are environments where that's not always true. So that's my plug. Research noise in the United States or not in Europe. Keep doing the Europe things and pay attention to the sort of patterning of air pollution and noise exposure.
And I was actually curious about that as well. You know, why is it? What are the obstacles here that make it so difficult to do those studies? Or maybe that's something we could talk about tomorrow. So I'm going to hand it over to Lucy. She's going to wrap things up, and then we can head on out for the evening. Thanks, Lucy.
Great, thank you. So the wrapping up, and those of you who are attending the workshop, you can drop off now. These are more logistic and organizational comments. I'm showing 29 attendees, but I only see 3. So I'm wondering if this is a technical problem on my end. No, people are just starting to drop off. So some of the things that I wanted to talk about very briefly are sort of what you would like to, as panelists, what you would like to get out of this workshop in terms of a work product, because it will help me to structure our discussion tomorrow at the end of the meeting. You know, we can generate a report based on the abstracts that you provided to us and, you know, the main points from your talk, and just have it be, you know, a summary proceedings of the workshop. Or we can do something that is a little more detailed and identifies gaps and research needs, and is a little more synthesis. And so I'm wondering what your thoughts and ideas are, and your reward for sticking around is that you get to provide input into this. So let me just stop there and open up the discussion and see what people's preferences are. Obviously, the more synthesis would involve more effort from the panelists. You would need to provide input into that.
This is Matt. I think I would favor something that's more synthetic. Particularly, I think tomorrow we have some things that are a little bit different from what we heard about today about particulate matter and exposure. Maybe tomorrow we'll be hearing more about the science of measuring the behavior that is related to exposure. How does the exposure affect the behavior? How does the change in behavior reflect the exposure and development of disease? That's a lot different than thinking about the basic science of inflammatory markers, how they're delivered through the nose, what portions of the brain they get to. Ultimately, they're related, but there are multiple steps in between. I think maybe we need to articulate that, isolate the different parts that maybe could be targets for investigation, and think more synthetically as you suggest.
Thank you. Others?
I like the idea of something synthetic that could bring the idea, somebody mentioned about the discussion was great. So if we can somehow capture some ideas in the discussion and connect the topics, even as Matt said, that would be great.
I think if we could get a little bit more into verification and validation, that would be helpful. Some of the stuff we heard about that's really important is kind of epidemiologic, and that's like history. And then, so how do we move forward with longitudinal studies, actually collecting data prospectively? Another thing that I heard today was the need to analyze huge amounts of data with lots of variables, and it's confusing and teasing one out from the other. And I don't think we have time for this tomorrow, but we need to talk about data analytic approaches. And even more, we need to talk about pathways analysis and theories. I didn't hear a lot of that today. And I think in order to make sense of a big ocean of stuff, you have to have theories and you have to have hypotheses around the theories. And I didn't hear that today.
Thank you. Anyone else?
I think, let me also say validation and verification. So when we heard about the Danish study, there's a lot of noise. It would be good if somebody went out there with sensors and actually validated if these are really noisy places. We don't know if they were. We don't know if it was noisy in the apartments where the people lived. So you can't go everywhere, but maybe they should do a reality check and actually go and measure what you think is going on, because maybe it's not.
Yeah, getting as to what Matt was saying, noise measuring and exposure assessment, particularly in this country, is in its infancy, real infancy, especially when we think of road traffic noise, something I'm going to bring up tomorrow. And so looking at the gaps and how do we fill in those gaps, I think is really important if we want to start looking at the exposome. And so I'm really interested, especially tomorrow, in people talking about how could we better do those measurements, and so that we can do the kinds of studies as Jennifer said here in the US.
Okay. Anyone else? The other question